RNA Collaborative Seminar Series. My name is Benoit Chabot. I've been involved with the RNA Society. I'm also a member of the RiboClub. Hopefully many of you know both activities. And today we'll be lucky to have two very dynamic representatives of the Canadian RNA science field. We will have two talks. The first one will be given by Dr. Hosna Jabbari from Alberta, and the second one by Julie Claycomb from University of Toronto. We will start with Dr. Jabbari, and I will ask right away that after their presentation, or even during their presentation, you're welcome to use the Q&A part of the presentation to put your questions in there, and I will eventually read them to the presenter after their presentation. This will speed up the process rather than having everybody individually asking their question, but you will be welcome to continue asking questions. And even if, for example, your question are not answered, I'm sure that the presenter will be happy to provide answers to your Q&A using the chat box. So I will start with Hosna Dr. Jabbari, who obtained her PhD at UBC, and then she got a PDF, postdoctoral fellow at University of Alberta. And then she spent a few years moving around. She went to Vermont, to Victoria, to Penn State, eventually returning to Alberta, the University of Alberta, where she has been since May 2025. She's been very active in the RNA sector, particularly in our recent activity in RNA Canada IRN. She's been a chair of the membership and analytics committee, and she's been sitting on the education committee as of the RNA Canada. Her talk is entitled RNA Structure to Function, and we're very excited to have you, Hosna, and it's all yours. Thank you.
Thank you very much for the introduction. I will start sharing. Okay. So my talk today is going to be an example and a story to show you how we can connect the structure to function, or how can we deduce function from a structure. When we talk about a structure, people have different understanding of the structure. We have three levels of a structure: primary structure, which is the sequence information; secondary structure, which is the set of base pairs. We are focusing on hydrogen bonding; and tertiary structure, the three-dimensional, the structure that ideally everybody wants to get to. The focus of my talk is mainly on the secondary structure, because with the set of base pairs, we can get a lot of information about the tertiary structure. At the same time, it's much easier to predict than the tertiary structure is. When we talk about secondary structures, we have two types of a structure, so two classes of secondary structure. The first one I'm going to call pseudo-knot-free. There are many different ways to present this structure. This one is hopefully a familiar case as a planar graph, but if you take the two ends of this RNA in your hand and assume that you can pull this sequence so that the sequence forms an arc, a line, and then base pairs are still intact, you will see this structure. In a pseudo-knot, so the arcs are the base pairs, and the sequence is on the line, on the horizontal line, and the 5' end is the base 1, and the 3' end is the last base. So if I want to define this better, I will say that I can show the same structure with dot brackets. Open brackets show the 5' end of the base pair, and close bracket the 3' end, and unpaired bases are shown with a dot. And mathematically speaking, for every two base pairs that you can find in this structure, they're either disjoint, like these ones, or they are nested, like these ones. Hopefully you see my cursor as well. The second type of structures that we have as part of secondary structures are pseudo-knotted structure. This is the famous structure of a kissing hairpin, which we have a hairpin over here, base pairing with unpaired bases of the hairpin over here. If we do the same thing and pull this structure apart, we will have the structure on the right, and the long-range interaction between the two hairpins is going to show itself with crossing arcs. The crossing arcs is the characteristics of pseudoknotted structures. When you want to show pseudoknotted structures in dot bracket form, you will need more than one type of bracket to show this crossing base pairs. And mathematically speaking, we call a structure pseudoknotted structure if you can find at least two base pairs such that they cross one another. So with that, I'm going to tell you the problem for secondary structure prediction. So the problem for secondary structure prediction is that you have been given an RNA sequence and you want to predict its secondary structure. Majority of the methods use an energy-based model. In this energy-based model, every feature has been assigned an energy value, and then you add these energy values up to get the energy of the whole structure. There are, of course, simplifying assumptions for that. One is that RNA forms a structure that has the lowest free energy. That means it's going to be most stable. At the same time, we are assuming that this RNA structure formation is going to be in isolation with minimal or no interaction with environment. The challenge, of course, becomes finding the structure with the minimum free energy among the pool of exponentially many structures that an RNA can form. If you allow for pseudoknotted base pairs, then the problem becomes infeasible, or as we call it in computer science, NP hard. And in this specific case, it's even harder than that because you can't guarantee an approximation for that either. That's for the computer scientists in the crowd. But you know that we have a lot of different methods that can predict a structure. Each one of these methods focuses on a specific type of a structure. So the famous M-fold algorithms of Zucker and Stigler, for example, only handle pseudoknot-free structures. It is relatively efficient. So for computer scientists, again, the time complexity is n cubed and n squared for space, where n is the length of the sequence. The most complex and comprehensive pseudoknot prediction method is Peanuts method by Elena Rivas and Sean Eddy. That is not very efficient. So you can predict the structure for complex pseudoknotted structures, but the length becomes relatively restrictive as about like 100, 200 base pairs only. We have different methods that are based on different hypotheses. I will talk about iterative H-fold and H-fold in this talk, and they can predict a complex pseudoknotted structures and are not as comprehensive and as the Peanuts method, but pretty comprehensive. I'm happy to answer questions about how comprehensive they are, of course. So now let's move to a known example. So we have all been affected by SARS-CoV-2, COVID, and one of the works that I can talk about moving from structure to function relates to our work to this specific virus. We all know that this is a single-stranded RNA virus, and the length is about 30,000 bases. At the time that we started this work in about 2020 or early 2021, we knew that there are some structural elements existed in 5' UTR in the frameshift element or the 3' UTR of this RNA. There were some experimental data, experimental work available with shape or with other methods, DMS specifically, and some computational methods that looked at the structures for the whole genome. But little was known at the time about the structure encoding region, and a lot of these structure predictions, either experimentally or computationally, didn't give us a comprehensive view of what structures are possible because they were not conclusive. So a PhD student of mine, Alison Ziesel, started looking at changing a known pipeline to predict the structures in the whole genome for SARS-CoV-2. The pipeline was previously used for predicting conserved structures in vertebrates or for non-coding RNAs, but we had to change this pipeline so that the criteria becomes more stringent, and we added pseudonaut predictions like cacofold to the structure prediction method, and we cross-referenced these findings with other methods. So over here on the right, you have the pipeline, and the pipeline starts with sequence collection. So the sequences that we collected were 13 different viruses. Most of these viruses were human affecting viruses. We had bat viruses included in a bunch as well. We had bovine. Bat was because it was quite similar to the original SARS-CoV-2. At the same time, we included MHV because there was a lot of data on this virus. And then we went through, created the phylogenetic tree, aligned the sequences, and created the structural alignments. We extended the structures and repredicted the structures for specific regions. And then we compared with available data. So based on this pipeline, Alison identified 40 regions in the whole genomic sequence for SARS-CoV-2 that we believe has function. So these 40 regions, a majority of these regions included conserved stems, like the one over here that we confirmed existence of a structure in a frameshift element. But there were some conserved regions like this one that were the conserved loop elements. To make sure that these structures are not hypothetical structures and they have functional value for the virus, we looked at variants of concern and we looked at mutations. The hypothesis is that if there was a mutation and the structure of any of these regions changed, then that means that was not functionally important. So Alison changed the pipeline, looked at the variants of concerns, aligned them, looked at the nucleotide count positions for the variants of concern. And if the mutation existed for 75% or more of the sequences collected, then we characterize that as an existing mutation. And we looked at the structure and we cross-referenced the structure with the 40 regions that we had seen before. So Alison identified these mutations on the virus. So the mutations were cross-referenced with the outbreak info. And in the outbreak info, so you see the variants of concern on the left. These are by the time that this work was done. And the horizontal line shows the virus, and top of the line shows the single point mutations that Alison detected, and the bottoms are the ones from outbreak.info. You may notice that we may have a lot of different single point mutations, for example, here and here, whereas the outbreak info has one mutation. This is because outbreak only considers that as one deletion mutation, but for us, we have multiple single mutations over here. The other thing you can notice over here is that we have both synonymous and non-synonymous mutations. So the synonymous mutations are the ones shown in white, whereas non-synonymous are the ones in black. And we couldn't find synonymous mutations in outbreak info. And we have also coding region mutations in non-coding regions, sorry. And when we cross-referenced this with the 40 structural regions that we previously predicted, we saw that no major structural change happens with any of these mutations on these 40 regions, giving us more confirmation that these structures are functionally important. So then we focused on one specific one of these structures, and that was the frameshift pseudonaut. Frameshift pseudonauts is important because in SARS-CoV-1, it was hypothesized that covering this frameshift element or reducing the yield for the frameshifting can become a cure for the virus, for the infection, and that was something that we were trying to see whether we can have something like that or we can help with something like that for the SARS-CoV-2. Frameshift is a key element in the application of coronaviruses in general. In SARS-CoV-2, we have two open reading frame, or 1A and 1B. 1B is out of frame with regards to 1A, so normally only one of them is being translated. However, there's a structure over in this specific region, shown with the red arrow, that facilitates moving the ribosome one base backward and creates a fusion of these two ORFs. In terms of sequence similarity, frameshift element in SARS-CoV-2 is very much similar to the frameshift element for the SARS-CoV-1, and it only has a single base difference. In SARS-CoV-1, people said that the functionality of the structure depends on the plasticity. Plasticity being the multiple structures closer in terms of energy to the native or minimum free energy, or the most stable structure. And these structures facilitate the function as well as the original structure. So that was the other thing that we were looking into for this specific structure. So a PhD student of mine, Luke Trinity, who's now a postdoc at Jackson Laboratory, started working on this. And in addition to finding or characterizing the structure and its pathway, he also looked at the possible mutations. We know that if the structure is functionally important, there are not a lot of mutations, so there weren't many. And none of these mutations is going to cause a certain structural change to limit the function of this structure. The way that we looked at this structure prediction for frameshift element was going with a hierarchical folding hypothesis. Hierarchical folding hypothesis mentions that a sequence, an RNA sequence, first folds pseudonaut-free structure once the ends of the sequence are closer to one another, then more complex base pairs that can possibly form pseudonauts forth. So there are two methods based on hierarchical folding hypothesis. One is H-fold, the other one is iterative H-fold. The difference is H-fold receives as input the RNA sequence together with a pseudonaut-free input structure and considers this input structure as a constraint, meaning it doesn't change, and only tries to add base pairs to lower the energy of the given input structure. Whereas in case of iterative H-fold, we allow for minor modification of the structure to accommodate more stable base pairing. So iterative H-fold receives a sequence and a pseudonaut-free structure just like H-fold, but can change that input structure to find more stable output structures. And it considers the input structure only as a guidance or a restraint here. So following or using iterative H-fold, we obtained the input sequence. We calculated energetically favorable stems. You can see over here, these stems are ranked based on their energy values. And then we fed these stems as restraint to iterative H-fold and predicted the secondary structure. The secondary structure, the first one, so these ones are also ranked based on their energy values. The first one is the same as the native structure for the SARS-CoV-2. And as you can see over here, the multiple initial stems can lead to this structure. So there are other structures that are energetically close, and you will see that one observation is that multiple initial stems can go to multiple of these structures. This is probably a confirmation to plasticity of this structure. The other thing that we noticed is that if this is the native structure, there are multiple structures that are energetically close, like this one. So if you consider blue and red, that's one structure. If you consider black and red, that's the other structure. They are energetically very close, but one of them is native structure, the other one is not. So we were thinking of understanding better what non-native structures are and what type of effect they can have in the function. So for to find the non-native structure, to look at the non-native structure and incorporate existing information, we looked at shape. For shape data, we have to, it's better to fine-tune slope and intercept. So we looked at previous data that was from shape knots. We added existing data to it, and we went through a grid search to find the correct slope and intercept. Then we used the 4 existing shape data sets to predict the structure for frameshift element. One other thing that we needed for prediction was that the method used should be very robust because, as you can see in this figure, not all of these shape data sets conform to one result. For example, in multiple places, 2 are showing a non-existent or no reactivity, whereas the others are showing reactivity for a specific location. So we modified our pipeline, obtained the sequence, and then looked at incorporated shape in addition to energetically favorable stems, and looked at Shapeify here to predict the structures. Predicting the structures, input structures are on the left, output structures are on the right. You can see that multiple input structures can lead to the native structure, and the darkness of these paths shows the consensus of these shape data informations. So as you can see, from stem 1 to the native structure, there is a high consensus. The other thing that you can notice is that we have multiple structures that are energetically close to native structure, and in majority of these cases, there are more than one stem that can lead to these non-native structure or native adjacent structures here. If we look at native non-adjacent structures, so structures that are farther away in terms of energy, you will see that this consensus between the shape data set is not as much as the native or non-native adjacent structures. And then we don't have multiple stems that lead to one structure. So these are probably non-functional structures. So then while we were working on this, other people identified more than one motif for the frameshift element, and these motifs were shown as 3-3 or 3-6 motif. To let you see what these motifs are, I'm going to introduce this formation, which is the dual graph for motif identification. In this dual graph, stems are the vertices, and the edges are loops. So over here, we have 2 stems, we have 2 hairpins with a self loop, and then another loop between the 2. So 2 hairpins and another empty loop between the 2. This is, again, two vertices, 2 stems, stem 1 and stem 2, 1 hairpin over here, and there are 2 loops between the 2 stems, so 1 and 2. And in case of a pseudoknotted, we have 3 loops between the 2 stems. So following this, we wanted to look at available motifs. To do that, we wanted to also open up our view and see whether these motifs can change by zooming in on the frameshift element versus looking at a wider area. And to be able to give a better comparison between the 2 motifs, we looked at the energy of the structure divided by the sequence length. We looked at a conservation. So on top of the line, you will see the conservation in different versions of coronaviruses, for example. And this is the attenuated hairpin, this is the slippery region, and this is the assumed pseudoknot interaction area. In the bottom, you have the native structure, and this conserved structure was predicted by Argonaute. So we looked at Shapeify for a longer window, and we saw that when the native structure is the 3-6 motif, we saw a lot of cases with 3-3 motifs, so the one on the bottom. And these 2 motifs are non-compatible, and they only share the dark blue stems, as you can see in the middle. So the red stems and the light blue stems are non-compatible. And we saw majority of the motifs showing 3-3 motif, whereas previously, everybody thought the 3-6 motif is the native structure. We looked at different variants of SARS-CoV-2 with different mutations, and we also saw that in 5 of the 7 cases, we saw 3-3 motifs. Then we looked at bat coronaviruses, and majority of those also showed 3-3 motif as the one that was more persistent or more bold. Then we generated about 11 1,000 sequences, and we looked at the various length for these sequences to see whether motif is different by length. So we know that there are multiple motifs that exist in the ensemble. We wanted to see which motifs at which state, at which length are going to be the most stable motifs. So around 100, you will see that the most stable motifs are 3,6, the native. But then when we go open up our view and look at around 110, for example, 3,3 becomes the most stable motif. And it continues until about like 140-ish. And then maybe around 150, 3,6 motif becomes another stable motif. And we looked at other motifs as well. So the question was, what causes this change in the structure, and how can we justify seeing the different motifs? We looked at the path convergence for 3,3 motif over here, and 3,3 motif seems to be a plastic, does have the plasticity characteristic as well. So stem 1 and 2 both can form these two motifs. So for that, we created a partition function method. And in this partition function, we looked at the ensemble free energies for these different motifs. And what we did was that we started removing one base from the 5' end of the sequence, and we looked at the changes in ensemble free energy. That was to resemble move an attachment of a ribosome to the sequence. As you can see over here, this was the full length. So this is the full length frameshift element. On top, you see the changes in ensemble free energy. And on the bottom, you see the changes in the structure. So assuming or enforcing refold, we have removed one base at a time from the 5' end of the sequence. And as you can see from the start, if this is the ensemble free energy for stem 2, and this is for ensemble free energy for stem 1, the ensembles are close to one another. And then after removal of the 5th base from the 5' end, the ensemble free energy from stem 2 in dark blue becomes less stable than the one that has initial stem 1. That possibly shows the flicking point between the two structures to make one more stable. So as you can see over here, we confirm that with our structure prediction as well, that once ribosome moves from 5' to 3' on the frameshift element, this structure changes. So after you get to this G, the blue stem is no longer stable enough. So the red stem wins the competition and makes the functional structure after that point. So that was the whole story for us. Hopefully that will help with other cases as well. This project was done in collaboration with Sebastian, Ulrike, and Jan. And this is a snapshot of my lab in about 2020 when this project was done. This is Luke, but we don't have Allison over here. Thank you.
Thank you, Hosna. Thank you very much. So we have a few minutes for question. I can see one in the Q&A, and I'm going to read it to you. What you presented looks great to predict new structures that have pseudoknots. Can it be adapted to homology searches like Infernal to find models that have pseudoknots? If yes, how will it affect time required to search structures?
Mm-hmm. So you can use Infernal for homology search, of course, but it's not that straightforward when you have pseudoknotted structures. If the pseudoknotted base pairs are kind of clear, then you can create a model based on the pseudoknot free part of the structure, look for that, and then manually search for possible base pairs that can form pseudoknots. If you do that, then you're going to be limited with the same time constraint as a pseudoknot free structure search with Infernal is going to take. So that's not going to take much longer, but it needs user input as well.
Thank you. So this question was from Jonathan Perrault. We have another one from Thomas Schneider. What were the units of energy? One at the scale of 0.01 and the other on the order of 20.
So the unit of energy was always kilocalorie per mole. and the structures that had similar energies with normally within the 2 kilocalorie per mole difference. And that was adjusted by, and when we looked at this different length, that was adjusted by the energy per length. So unit of energy per nucleotide.
Okay. Now, I can maybe have a question from a non-structural biologist, because I was puzzled by, I mean, for the SARS-CoV-2, for example, there's so many possible local structure. And I'm just wondering if there's, I mean, sometimes maybe a structures need to be transient. And this, could this be featured by, for example, having the ability to fold a particular region in 2 very similar energy mode, and then so that the time spent in each one is sort of lost or shared, such that this may actually help the virus in a way, because I mean, the virus has to escape this double-stranded RNA monitoring system that will trigger the interferon response and kill the cells. So maybe the virus has learned to escape. Could it be a mechanism by which the virus has learned to escape that monitoring in a way, by having structures that are rapidly exchanging between one another?
Yeah, so we didn't think about that in this specific project, of course, but something I can say is about the motifs that we consider. So if you look at the ensemble of motifs that we had in our slide, about the 11,000 of them, there were multiple structures that existed, and some of them were as stable as the other ones, specifically to some extent around 150-ish, 140, 150 nucleotide base. You saw there was a competition between red, blue, and sometimes green dots, right? So that showed the structures that were close to one another. I can add that because our energy parameters or energy models are simplified, we do not have an exact value for these structures, and we do not consider other interactions that these structures might have. So while we indicate that these structures are energetically similar, one of them might be preferred by the situation with the environment than the other ones. But what we can see is that there are multiple structures that have close enough energy that either one of them can be functional.
Right. Okay. Thank you very much. And we don't have any more questions. The timing is perfect. You did your 30 minutes perfectly. Thank you very much, Hosna, for your presentation. So we're ready to move to our next presentation, which will be done by Julie Claycomb from University of Toronto. Julie has done her PhD at MIT, and then did a postdoc with Craig Mello at UMass Medical School. She's been at U of T since 2011. She's now vice chair of the Department of Molecular Genetics. She's been very active in the RNA field in RNA Canada. She's been secretary of the executive committee and director on the board of RNA Canada, and also very active in the RNA Society. She's been a board member on the RNA Society. And for those who are not aware of it, I'm happy to tell you that Julie has accepted to be the co-lead of the RNA Society meeting that will be hosted in Montreal in 2026. So thank you very much, Julie, and the floor is yours. Thank you.
All right. Thanks, Benoit. Thanks, everybody, for being here. I saw lots of friends and colleagues out there in the audience, so I really appreciate you being here today. And yeah, we hope to welcome you to Montreal in 2026 for the International RNA Society meeting. Okay, so today I'm going to tell you about a project that's ongoing in the lab, and we're making nice progress on it by a talented PhD student, Sebastian, who's here on the left, and our wonderful research associate, Samantha, who the two of them have been working on this project to bring it to fruition. So my lab is working to understand the mechanisms of small RNA-mediated gene regulation. We use a variety of approaches. We use cell biology, genetics, genomics, molecular biology, And all of this is tied together in our trusty little worm, C. elegans, which is a fantastic model for understanding small non-coding RNAs. And maybe you've been reminded of this recently, that worms have been a leading champion for small RNA research for many, many years. So for instance, microRNAs were discovered in worms a few years ago. We've all been reminded of that this fall. Double-stranded RNA was determined to be the active agent in RNA interference using worms. That was the work of my postdoc advisor and Andy Fire. And these proteins, Argonauts, which are the core effector molecules in these small RNA pathways, were first linked to microRNAs and RNAi by some beautiful genetic screens that were done, well, about 25 years ago now. So, as I said, the Argonaute proteins, or the AGOs as we call them, are the engines of small RNA pathways. And here's the crystal structure of human Argonaute 2, solved by a few labs. This one is from Ian McRae's lab. And so here you can see the Argonauts got these prescribed domains, and you can see part of a microRNA that's in association with the Argonaute. And Argonauts can do a lot of jobs to regulate gene expression. For some of them, they have the capacity to act as endonucleases with some slicer residues that are down in this region. So they can directly cleave and degrade transcripts. They can promote RNA decay by recruiting other factors like decapping enzymes and deadenylases to do the dirty work of degrading that RNA. They can inhibit translation by interacting with translation components in the ribosome. And they can inhibit transcription by bringing into with a nascent transcript histone modifiers that induce heterochromatin formation, or actually directly attenuating RNA polymerase 2. On the flip side, so those are the silencing activities for the Argonaute that we usually think about and associate with small RNA pathways like RNA interference and microRNAs. But on the flip side, there have been a few sort of positive or licensing activities that are elicited by small RNA pathways, in which Argonaute binding can actually stabilize transcripts, or it can promote transcription by inducing euchromatin at target loci. Right? So Argonauts are diverse silencing and sometimes licensing machines. And what you may or may not know is that while C. elegans is a great champion of RNA biology, small RNA biology, it's kind of a pain in the butt to study small RNA pathways in, because there are actually at least 19 functional Argonauts in C. elegans. And so recently, my lab set out to understand what the small RNA binding partners and functions of these Argonauts were. So we had this systematic study where we actually used CRISPR genome editing to introduce GFP and 3X flag tags into each endogenous Argonaute locus. And with that, we could look at the expression and localization patterns of these Argonauts, or we could use antibodies against those tags to pull down the Argonauts and do high throughput sequencing on those Argonauts to find, on the small RNAs associated with the Argonauts to find what the targets might be computationally. On the flip side, we had some classical mutants of these Argonauts, mostly deletion alleles that took out parts of the protein coding region, but we also generated some designer alleles with CRISPR genome editing. And with these different null alleles of these Argonauts, we could look at what went wrong with the worms when we lost the function of those Argonauts. So we did all kinds of phenotypic analyses, and we also looked at what happens to the small RNA populations in total when we remove one of these Argonauts. Is there a type of small RNA that's lost, and does that correlate nicely with the Argonauts that were associated with that Argonaute with the small RNAs that were associated with that Argonaute from the IP small RNA sequencing? So we've got all these beautiful data. It's a nice foundation for our field. And what I want to home in on today is the expression and localization patterns, because they've informed a lot of what we've done subsequent to this paper. One of the things that we found was that most of the Argonauts, 16 out of 19, are expressed in the germ line. And so the germ line, as you know, is a really important tissue for ensuring the survival of species. And in C. elegans, we think of the germline as an immortal cell lineage. And in this cell lineage, we start with these two primordial germ cells that proliferate to make the gonad here. And in an earlier stage of development, the worms, these are hermaphrodites. So they'll first make sperm, they'll undergo spermatogenesis, and then remarkably, in those same gonads, they'll switch at the adult stage to making oocytes. And they've stored up those sperm in the spermatheca, and they can fertilize these oocytes as they pass through the spermatheca here. And from the very first cell division, the worm sets aside a cell that has the potential to build a new germ lineage. Okay, so from the beginning, and there's this continuous cycle of the germ cells. And one of the things I want to highlight here, maybe you can see them, are these little green dots. And these little green dots are what we call germ granules. And they're really important for germ cell function and the maintenance of fertility, because they carry germ cell determinants, so determinants of the cell fate lineage. They have important roles in small RNA pathway functions, and they contribute to this really cool process of transgenerational epigenetic inheritance, which worms are a really great model for. And so in C. elegans, it seems like, so we've got 4 germ granule compartments that we can study. And it kind of seems like there's a new one every week when you look in the literature. So I'd say we're up to above 4, maybe up to about 7 at this point, but I'm going to talk about the 4 main ones that have been studied really robustly up to this point. And so over 40 years ago, the P granules were discovered. These are shown here in green. And they've been the most well-studied over the years, mainly because, well, they're large and they're well-known, and they were identified quite a while ago, so there's been time to study them. About 6 years ago, the Z granules, which are associated with the P granules, were identified, and they've been studied, and they're involved in transgenerational epigenetic inheritance. Mutator foci are the ones shown here in this whitish gray color, and they've been known for about 10 years, and they're involved in the amplification of small RNAs, so the biogenesis of a major class of the small RNAs in the worms. And finally, we've got these simmer foci, which were discovered only a couple of years ago, which are thought to aid in the production of those small RNAs in a slightly different way than these mutator foci. And so while we, and so here, I'm just going to show you what this looks like if you look at it under the microscope. So again, in green, we've got the P granules. In red, we've got the Z granules. In white are the mutator foci. The simmer foci aren't shown here. And below this would be the nuclear pore. And if you've ever heard me give this talk before, you'll know that I think this looks like the Grinch, and it's almost Christmas time, so it's totally appropriate. So this is the organization of these components. And again, the transcripts will leave the nuclear pore. They'll transit through the P granule, and then they'll be routed to these other granules, possibly for small RNA biogenesis or other small RNA regulatory functions. And so, like I said, we know varying amounts about each of these different types of granules, including the numbers of proteins that make up these granules. And so for P granules, we've studied them the most, and we know over 50, I think at this point it's probably over 75 components that are involved. Z granules, we've only got a handful of components. Mutator foci, about a dozen, and these simmer foci, there are only 2 known components at this point. And so loss of these germ granules, as you might imagine, leads to sterility, loss of germ cell fate, and defects in the ability to do RNAi and inherit RNAi. So they're really important for the function of the germline and for small RNA pathways. And so, like I told you, 16 of those Argonauts localize or expressed in the germline. And what we noticed was that 8 of these Argonauts actually localize robustly to germ granules. And here's what here I'm showing you some of these Argonauts. What you can see is a single nucleus here, and the Argonauts are tagged with GFP, so they're green, and the Z granule component ZNFX1, it's a helicase, is shown in red. And you can see that these Argonauts overlap to varying extents with the Z granules. And so we obviously wanted to know which granule does each Argonaut associate with. And we also wanted to know, what are their functions? What are these Argonauts doing within these germ granules? Are these just storage areas, or is there actually some gene regulatory function that's occurring here? And as you might guess, this part's hard to figure out. And so instead, we turned our attention to understanding what the Argonaute protein-protein interaction network might look like in these granules. And this is a little bit tricky because you can't actually purify these granules intact. They tend to dissociate upon purification. They're phase separated. And so we turned to a different approach to understand what the Argonaute protein interactome might be in these granules. And so we used proximity labeling with Turbo ID, and we took two different approaches. In one approach, we directly introduced the Turbo ID enzyme into the bait, the Argonaute protein, as a translational fusion. And in a second approach, we used those GFP-tagged Argonauts and we crossed them into a different strain that expresses a nanobody. And that nanobody has the Turbo ID enzyme attached to it. And it's a nanobody against GFP. So it'll recognize that GFP on the Argonaute, bring the Turbo ID enzyme in proximity. And what Turbo does is basically nearby proteins will become biotinylated because Turbo activates biotin. And so what you get is this cloud of biotinylated proteins nearby your protein of interest. For the direct Turbo, the rule of thumb is 10 nanometers, but that's the really rough estimate. And so what we're identifying here are proteins that may be direct interactors and or in close proximity to those Argonauts. And we do pull downs with streptavidin and subject these to mass spec. And so as a disclaimer, if I ever say interactors throughout the rest of this talk, what I mean is proteins that are in proximity to these Argonaute baits. They might not actually be direct interactions, and it's important to remember that. All right, cool. So we initially focused our efforts on 4 Argonauts because 8 Argonauts was a lot for a new PhD student to jump into. And Sebastian has performed admirably in doing lots and lots of mass spec experiments here. And so we focused on WEGO1, WEGO4, Caesar1, and PRG1. And these Argonauts do different things. So WEGO4 and Caesar1 actually bind to the same subset of small RNAs in the worm called 22G RNAs, and they target about 5,000 germline protein coding genes that are really important for the germline. Okay, so binding the same small RNAs, targeting the same genes, but they act in very different ways. WEGO4 silences its target genes, and Caesar1 licenses, or it promotes their existence and expression. So they're acting in sort of counteracting ways. And so we wondered if one of the ways they could behave in such different capacities was by having different protein interactomes. Next, we looked at PRG1, which is the main PWE, the lone PWE expressed in C. elegans. And PWE binds to pi RNAs and is on surveillance duty for all kinds of foreign nucleic acids like transposable elements, transgenes, etc. And when PRG1 finds these foreign nucleic acids, it calls in WEGO1, which is one of these worm-specific Argonauts. That's where they get the name WEGO, worm Argonaut. And WEGO binds to a different set of small RNAs, also called 22G RNAs, but to different genes. And WEGO1 silences these invading transcripts, as well as some endogenous protein coding genes downstream of PRG1. So PRG1 and WEGO1 tend to work together to reinforce each other in silencing activities. Okay, so we did these Turbo ID experiments, and what Sebastian found was that Caesar1 and WEGO4 seem to have 2 totally different protein interactomes. Even though they're binding to the same small RNAs, they're doing different things, and they have different protein interactomes. So maybe this wasn't surprising. WEGO1 and PRG1 are functioning together in a 2-step sort of pathway, and they showed us a largely overlapping protein interactome. And so, oh, sorry, let's go back. Wrong direction. Okay, there we go. So let's focus in on some of the things that we found in our protein interaction networks. So for Caesar1, one of the interesting sets of components were the nuclear pore proteins. Okay, so we found a lot of nuclear pore proteins that are on sort of the basket side, the cytoplasmic side of the nuclear pore. These are a lot of FG rear P proteins, and they're probably involved in anchoring the P granules. Okay, so Next, we focused on WAGO4. And one of the things that we noted that was distinct about the WAGO4 protein interaction network was that it contained a lot of helicases, as well as RNA degradation enzymes, which could explain its silencing activities, and some other Argonauts that it interacts with and may be cooperating to regulate a common set of transcripts or something that we're not sure of yet. Turbo ID on WAGO1 and PRG1 show that shared proteome, and some of the things that we noticed there were pRNA biogenesis factors, 3' end-tailing enzymes, and structural components of germ granules. And so we took a step back and overlapped our Turbo ID data with other existing P granule and Z granule component Turbo ID data sets to identify a core set of germ granule components. And so here you can see this hairball network. It's an absolute mess, but what I hope you can start to appreciate is that there are distinct protein interactomes for each of these different baits. And there's sort of this core set of interactors, which was really interesting to us, and we wanted to study that further. And so this core set of interactors was interesting because there are lots of uncharacterized proteins there, things with letter and number names like F56D 8.7 or something like that, nothing really intuitive. And so we honed in on a set of these uncharacterized proteins, and we've decided to call these guys AIPs, Argonaut interacting proteins, and you can see a few of them here. We've chosen about 8 of them to study. So we've got 7 AIPs, AIP 1, 2, here's 7, 3, 4, 5, 6, and a protein that was recently published, recently characterized, known as HERD1. And so these uncharacterized proteins are really interesting because they don't have a lot of remarkable domains that are RNA binding domains. So basically, we think that we've got a lot of novel biology to sort out here. But one of the things that's in common with most of them is that they have these intrinsically disordered regions or low complexity regions, which may or may not be relevant to germ granule biology. Although we do know that lots of proteins with intrinsically disordered regions are critical for phase separation and RNA binding. And so these uncharacterized proteins, we wanted to know, what do they do? What is their function? And so we took an approach where we knocked them down by RNAi and asked if their loss leads to fertility defects, germ granule distribution disruptions in the size or number of the germ granule components for P and Z granules, whether there's an RNAi deficiency. And so here we've got robust assays where we can feed the worms RNAi against GFP. And we can see this knockdown in the first generation. And remarkably, worms have this capacity to transmit RNAi knockdown for 4 to 5 generations without an additional RNAi double-stranded RNA input. And so that RNA inheritance is a really great assay to understand transgenerational epigenetic inheritance. And we wanted to know whether that could be disrupted when we lost these new putative germ granule proteins, these AIP proteins. And finally, we want to know if they localize to granules, which is still a work in progress, and I won't talk about that today. So let's look at fertility. And what we found was that knockdown of 4 factors leads to a reduction in brood size. And so here are the number of live progeny when we knock down each of those new proteins versus the empty vector. And worms usually have about 350 embryos, live progeny. And what I think you can appreciate is that a lot of these guys, nothing happens when you knock them down. But for several of these factors, you see a significant decrease in the overall brood size. So they are likely to be involved in fertility. Next, we looked at whether the germ granules were disrupted in any way. And we looked at 3 major things here. First, we looked at the distribution of these germ granules. And what we see is in blue, the distribution of a P granule marker relative to a Z granule marker. And in red or orangish is this the distribution, the overlap in pixels of this protein ZNFX1, which is in the Z granules, versus PIGO1. And in a wild type situation, the P granules are bigger and the Z granules are smaller, but more of the Z granule overlaps with the P granule. And I think that's what you can appreciate here. P granule is bigger, doesn't overlap as much with the Z granule, but the Z granule is smaller and overlaps more with the P granule. And we have a few of the factors that when you knock them down by RNAi, they look just like wild type. In some instances, though, we saw that there was an inversion where there was more overlap with the P granule marker with Z granules than with the Z granule marker with P granules. Say that 5 times fast. And so here you can see that. Here's the P granule overlap. It's an inverted pattern from what you see in wild type, okay? And then finally, we looked at situations where there was an equal distribution, where the P and Z granules sort of demixed and overlap with each other to equal extents. And we can see that here for 4 different factors. So that's interesting to us as well. And we'll follow up with these findings with more in-depth assays in the future. And then finally, we checked whether knockdown of these factors led to alterations in the inheritance of RNAi. Just to say this one more time, we can knock down GFP with GFP RNAi, and then after a few generations, it stays silenced, even without an additional RNAi input. In mutant situations, though, we may not be able to silence the GFP in the first generation, and that would be an RNAi defective mutant or RDE mutant. But we may also have differences in how long we can inherit the silencing effect, okay? And so that's what we were looking for here, differences in how long inheritance lasted. And in some situations, it could be shorter. In other situations, it could be longer. And so that's exactly what we've seen, is that some of these factors, when you knock them down, just look like wild type. However, some, when you knock them down, lead to shorter inheritance, and others lead to lengthened inheritance. So sort of taking the brakes off of silencing and letting silencing persist longer, which is a pretty cool phenotype. And so we do see that these factors appear to be involved in RNAi inheritance. And so just to sum it all up, what we've seen are that some of these factors have multiple phenotypes: fertility, germ granule defects, and RNAi inheritance. Some have just defects in fertility and RNAi inheritance, and some have fertility defects in just, and defects in just the germ granules. Okay, and so we'll be following up with these further to understand more what their functions are in the germ granules. So like I said, work in progress. So just to put a period at the end of this sentence, let me say that TurboID is a viable approach for identifying Argonaut interactions in these germ granules and in the germline. Mapping the germ granule proteome reveals new factors involved in germ granule biology and fertility. There are a whole bunch more uncharacterized proteins in those data sets that I showed you that we're excited to follow up on. And so again, I'll just thank the people who did the work. Adam Sunby was a master's student who started this work a few years ago at the beginning of the pandemic. And then Sebastian Fuentes came in as a PhD student and has spearheaded the project, along with Dr. Samantha Del Borello, who's our awesome research associate and keeps the lab running. She's my right-hand person. So thanks for your attention, and I'm happy to take any questions. Let me turn this off.
Thank you. Thank you very much, Julie. It was fascinating.
Thanks.
There is a question by Drew Galls. I'm so sorry if you said and I missed it, but have you done any of the fertility experiment over multiple generations?
Oh, that's a great question, Drew. So we did this a little bit, but not systematically. And so this is one of the things, what we really need to do, we used RNAi to knock these genes down, and that was a quick approach to see if there were any phenotypes that we might be interested in. Obviously, we really need to have, proper genetic mutations to study these effects and to look at whether there's a transgenerational fertility defect. And we'll do that both at 20 degrees and 25. We saw a little bit of it when we kept them on RNAi for a continued, for multiple generations. But I'd like to follow that up with proper genetic mutants.
Good. So we have another question here by Jennifer Porat. Wonderful talk, Julie. From the small RNA sequencing of the AGO knockout or mutants, do you see different steady state snRNA level for the licensing versus silencing AGOs?
It's a good question, Jennifer. Thanks. It's nice to see you out there. So what we see is that, yes, the licensing Argonauts, well, okay, I can't say it exactly that way. Caesar-1 and WEGO-4, I told you they bind to the same small RNAs and target those same essential germline genes. those two Argonauts have lower overall levels of 22G small RNAs against their targets than the other WAGOs, like WAGO1 or any of the other, WAGO group of Argonauts. So there's a difference already. And those WAGO group of Argonauts, we definitely think are silencers. We just don't know how, if it's like, post-transcriptional level, like at the level of RNA stability or translation. So there are differences amongst the different Argonaut small RNA pathways in general, in terms of the overall steady state levels of the small RNAs. And yeah, I think that's an important and interesting thing that the worms are trying to tell us, but we don't totally understand what that means for the biology yet. It's a really good question. We should talk about it more some other time.
Okay, we just want, oh, yeah. What control, from Sarah, I might have missed it, but what controls did you employ in the proximity labeling experiments?
Great question, Sarah. This is hugely important with those turbo ID experiments. So one of the controls that we do is always a wild type worm, and we subtract out, there's some really, there's frequent flyers that we definitely see in the data. We subtract those out with the wild type, and then we always use the nanobody alone control, and also we look at enrichment over the nanobody alone control to get what we think are the true interactors. We also do this Saint analysis. Our collaborator is Anne-Claude Gingras, who has developed the Saint algorithm, and so we have a pretty stringent pipeline for what we call as an interactor. And I am going to venture that we're missing more, that there are more things out there. We're being pretty stringent about it, and there are more interactors that we're not capturing. Great question. Thanks.
All right. I have one question myself, Julie. So
yeah,
the impact on one of the knockdown on the persistent RNA interference. So I don't know much about this pathway, but what other proteins are connecting to this pathway, and how do you see interaction? How do you see the interaction with these other candidates?
Yeah, it's a really good question. So these Z granules, actually, the component ZNFX1 and WAGO4 are involved in RNAi inheritance. And so it makes sense to me that we're going to capture some of these things that are both stimulating inheritance. I thought we'd get more of that. Like if you lose these things, you don't get inheritance. That's the more common phenotype we see in the field broadly. And we were surprised that we got these things that increased inheritance and are clearly acting as brakes. So, it's maybe not surprising that we see the thing that decreases inheritance. It's interacting. I'm not sure exactly which proteins it's interacting with, but we did the turbo on ZNFX1 and WAGO4, and it won't surprise me. I think it's in both of those data sets. Sebastian's probably yelling at me out there. Of course it is, Julie. So that link it makes sense to me. The extended RNAi inheritance, I'm not sure what to make of them yet, and it's going to take more study to figure that out. And we will bootstrap our way through these new factors and do turbo ID on them, and put them through the gauntlet of assays too. Yeah, sorry, it's not the best answer to your question. I see one more question in the chat.
Yeah, one quick question.
Oh, yeah.
Did you consider using split turbo ID?
That's a good suggestion. Yeah, split turbo ID to reduce background. That's a great suggestion, Luis. Yeah, we haven't considered it yet, but I think, yeah, it's something that we should consider. Thanks. Thanks for the suggestion.
Okay.
Okay.
And that concludes the question period. I mean, we cannot be more than 30 minutes. Both speakers gave exactly 30 minutes, including the questions. So I think you did both an excellent job. Thank you for your very, very nice presentation, both of you. It was a great pleasure to listen to you. And if people have more questions, you can still have a few minutes to write them up on the Q&A. They may still be answerable for a few more minutes. And let's end by thanking our speaker. I've seen a lot of clapping virtual clapping going on. And we can mark your calendar for the next meeting of the RNA Collaborative Series Seminar, which will be December 18. And this one will be hosted by the NCI Biology Initiative in Frederick, Maryland. So thank you very much. Have a very pleasant evening, and thank you for your presence, and see you next time. Bye-bye.
Thanks for being here, everybody.
Yeah.